Hi, everyone, and welcome. Today I will be discussing microbial testing and understanding the various techniques and method development that is associated with these analyses. And I do want to start by saying that some of the information I present today may be similar to what you have already heard, but in case we have some people joining the symposium late, I want to make sure that I review some of those basics before diving into method development and the different considerations that must be taken when this process is being carried out. For microbial testing, there are three prominent techniques that are utilized by cannabis laboratories. The first is going to be culture-based plating methods. The second is PCR or polymerase chain reaction methods. And the last is culture-based most probable number methods or MPN methods. And over the course of the next few slides, we're going to briefly review each of these techniques and discuss the analytical processes that are associated with performing each of them. The first method that I would like to discuss is going to be culture-based plating methodologies. And really for this type of method, what the laboratory is doing is they are attempting to grow organisms on a specific media in order to determine an overall count or even just the presence of the microorganism that is attempting to be identified. And in this process, what the laboratory is going to do is they're going to weigh up a specific amount of sample, and then they're going to add the appropriate buffer needed to grow that microorganism of interest. And they use the amount of sample that they weigh and the buffer that is added to determine an initial dilution. And from this point, the laboratory creates three stereo dilutions, and those are plated based on the requirements of the agar or the plate being used. And with this, I want to put some emphasis on the running three dilutions if it's a total count test being measured. For example, total yeast and mold or total aerobic count. There is some evidence that suggests that if you run too low of a dilution, you have some suppression happening due to oversaturation, so your results are suppressed or look lower than they actually are. So it's really important to do this so that you can get that average and really see what's happening across all of those dilutions. So once that sample has been added to the plates at the various dilutions, the sample is incubated for a specific amount of time, and then any microbes that were present in that sample will begin to grow. And at the end of that incubation period, the laboratory is going to determine if there are any colonies present, and if counts are required, for example, for total yeast and mold, then they're going to count the total colonies on each dilution and use the average to determine that final concentration of microbes that are present. And theoretically, the number of colonies should decrease as the dilutions get bigger. And in this diagram, you see here that that first plate has 16 colonies, the second has 8 colonies, and the third plate has 4 colonies. So if the assay is working correctly, this decrease indicates that the serial dilutions are increasing by a factor of 2 from one plate to the next. Again, I emphasize if it's working correctly because of that oversaturation leading to suppression in some of those plates. So you have to be careful and you have to examine each of those plates and understand what's happening with them. The next thing that I want to talk about with culture-based plating methods is the fact that there are different types of plates, medias, and or agars that can be used. And on this slide, I've listed out some of these plate types. So first up, we're going to have agar plates, and these are sterile petri dishes that contain a growth medium that has been solidified with agar and in some cases other nutrients and or antibiotics. And the four most common agar plates and the ones typically used by laboratories include DRBC, Sabdex, PDA, and PDAC. Next, we're going to have our Petri film plates, and these are going to be plates that are pre-made that contain the culture medium, a cold water soluble gelling agent, and an indicator that is going to assist with the enumeration of the colonies that grow. And for this type of plate, there are specific plates for the different microbes of interest. So for TYM specifically, there is a rapid and a non-rapid plate. And the rapid plate is going to result in colony growth in a shorter time period, while the non-rapid plate needs a longer incubation period to see that colony formation. The final plate type I would like to discuss is going to be the membrane filter plates, which are smaller petri dishes that contain a nutrient pad that has the proper culture medium present that has been dehydrated. And when the sample is added, it will evenly disperse across the plate, rehydrating that media that's needed to successfully grow the microbes that are present. And though there are several different types of these plates, one of the main ones available is referred to as a compact dry plate. And I want to say one other thing about this, and it's realizing that the different plates may need different growth medias or even initial dilutions, and they require different amounts of sample to be added to plates. So for example, those agar plates, which we discussed first, they can only have 0.1 mils of sample added to them, while the Petri film and the membrane filters have 1 mil of sample added. So that has to be taken into consideration when factoring in those dilutions so that you're properly quantifying the amount of microbes that you get if you're doing a total count test. The next method that I would like to discuss is going to be PCR, and specifically in this case, quantitative polymerase chain reaction, or qPCR, which is going to involve isolating the DNA of interest and then mixing it with primers to amplify the segment of interest in order to identify the microbes of concern. So whatever microbes you're looking for, whether that be Salmonella, Shigatoxin E. coli, Aspergillus, yeast and mold, aerobic bacteria, whatever it may be, you're able to identify it based on the DNA that's present. But before you can perform that qPCR analysis, the DNA from that sample must be isolated. And so depending on the methodology or the vendor, that extraction and isolation protocol is going to vary, but ultimately what you're doing is you're going to take a specific amount of sample and weigh it and add a specific amount of buffer. And once that sample and that buffer have been mixed really well and homogenized, the cells are going to be lysed so that any of the DNA that's present, both cannabis, microbial, whatever DNA is there, is going to be accessible. And so once those cells are lysed and that DNA is accessible, it's going to be isolated and separated from the sample. And then that purified DNA is then going to be used for the qPCR analysis. And that purified DNA is going to include all of the DNA that was present in that sample. So once that DNA is purified and isolated, qPCR can be performed, so the analysis portion. And for this, a master mix is going to be created that contains primers that are specific to the analytes of interest. So in the instance of running a cannabis sample, you may have a primer for something like Salmonella or Shigatoxin E. coli or Aspergillus, and then you're also going to have a primer for cannabis DNA. And the primer for cannabis DNA is present as an internal control, so it's going to show that your extraction worked. So you have both of those primers, you mix them together, that master mix is prepared. We then take our purified DNA, add it in, it's going to be mixed in at a specific concentration, and normally this is done on a 96 well plate, like what I have in the diagram, and then that sample is going to be, that mixture is going to be homogenized, and once that is homogenized, the plate is sealed and then loaded onto the PCR instrument. And once loaded onto the instrument, that instrument's going to be allowed to warm up, and then the instrument will begin undergoing cycles of heating and cooling. And as this happens, polymerization is going to begin to occur between the DNA that is present and the primers that were added to the master mix. So if the DNA segment that matches those primers is present, what's going to happen is it's going to bind together and begin reproducing exponentially. And this process continues as you go through those cycles. The polymerization then results in probe degradation, and this probe degradation allows a fluorescent signal to be generated and the DNA to be amplified. And the absorbance of this fluorescent signal at the end of the run is going to allow the concentration of the microbes present to be measured and identified, and in some cases even quantified. And if it is quantified, that quantification is done by conducting studies to show the relationship between that fluorescent point and the number of microbes that are present in a sample. Finally, we have the most probable number, or MPN methods, and this method is also a culture-based method. However, the principle behind how it works is slightly different since the colonies are not actually being grown when using this method. Instead, of looking at those individual cells quantitatively, what the method is doing is it's relying on characteristics such as turbidity or acid production, and it's using that to predict the number of growth units of microbes that would most likely exist if colonies were grown. In other instances, there's also most probable number methods where a substrate is used to produce a fluorescence. And in this method, you're doing serial dilutions just like you would with plating in order to confidently measure the microbes of interest. So when we apply this method directly to cannabis, it is going to be just like the prep for the other methods, in which a sample is weighed up at a certain amount and then a growth media or buffer is added to create an initial dilution. And from here, a secondary dilution is going to be prepared in order to remove interferences that may be present. And this could be something as simple as color or as complex as the product having a slightly acidic pH. The nice thing about performing this method nowadays compared to the past is there's actually a product available that will allow the serial dilutions to be created automatically by having a single card with a certain number of wells at various sizes. And as you can see in this diagram, you have 3 sets of 16, and each of them have a different size well, one being a small set, another being a medium set, and then the last being a large set. So once that secondary dilution that contains the sample in the buffer has been created, it can be loaded into that card, and then it's placed in the incubator for a specific amount of time. And over that time period, any microbes that are present will begin causing a physical change or a fluorescence to occur, which allows those microbes that are present to be measured and to be quantified. And as you can see in the zoomed in version of the card on this diagram, 5 of those large wells, 8 of those medium wells, and 8 of those small wells underwent a physical change that indicates the microbes are present in those wells. And using that information in a conversion to go from this most probable number to colony forming units per gram, or CFU per gram, the laboratory can determine how many microbes were present in that sample. Now that we have reviewed the various techniques, I want to talk about selecting the appropriate technique for testing and what some of the considerations are that surround this. The first is going to be live versus dead cell detection, and this is going to be particularly important with deciding between a culture-based method or a PCR method. PCR methods look at all of the DNA that is present, regardless of whether or not that DNA is dead or alive. Therefore, if you run a PCR-based assay, you may see higher amounts of microbials present, and that's simply because the assay will be able to identify any and all microbes that were present at any point in time, unless the DNA is fully destroyed. And the argument has been made that this can lead to false positives, especially for quantitative analyses such as total yeast and mold, where the total yeast and mold may have been present at some point, but now it's dead, so the hits shouldn't be a concern. And this comes into play a lot for a product that has been remediated in some manner, as the remediation should result in the death of the microbes. However, it may not lead to the destruction of DNA, which may explain some of the discrepancies that are being seen from laboratory to laboratory or from method to method. The next factor that must be considered is going to be the colony growth potential, and this is really going to be important for all techniques. However, the most issues associated with this are when culture-based techniques are utilized. So if you have ever worked in the cannabis industry at any point, you know that everything tends to be rushed, and clients want results out as quickly as possible. The issue with quick results for microbials is going to be that microbes take time to grow and form those colonies. Therefore, if that analysis is too rushed, there really is a chance that a false negative may be reported. And this is an argument that has been made for microorganisms such as aspergillus that can take 5 to 7 days to grow on plates, however, can be processed via PCR in about 24 to 48 hours. The final factor that I want to talk about when deciding which technique to use for microbial detection is if the analysis is quantitative or qualitative. So for traditional plating methods, the number of colonies that grow is equal to the colony forming unit per gram, or CFU per gram, which is how the regulations define the contamination limits. However, for PCR and most probable number methods, a conversion is done in order to determine the amount of colony forming units per gram that would be present based on the results obtained via each of those techniques. So with this, arguments have been made that traditional methods are best suited for quantitative analyses, since they're going to provide results that are not based on comparisons to another technique. They're going to provide results that are a direct relationship to the quantitation. Now that we have reviewed the different techniques and some of the considerations that should be used when selecting a technique and what goes into those techniques, I would like to review some of the method development considerations. So when developing a microbial method, there are several different factors that must be assessed. And some of these include things like inclusivity and exclusivity, detection limits and assay sensitivity, matrix validation, sample size, and inoculation requirements. And if you're attempting to develop a method that is AOAC certified, these, along with some additional requirements, must be met. And the specific requirements for each of these methods can be found in the standard method performance requirement, or SMPR, for a specific analyte. So there's an SMPR for salmonella, there's an SMPR for aspergillus, there's an SMPR for Shiga toxin E. coli. And so that SMPR will provide you with that guidance. And While I would like to touch on each of these today, I don't think time is going to allow for that. So instead, we're going to talk about the first three, as they tend to be some of the more challenging aspects of method development considerations. The first part of method development that I would like to review is going to be inclusivity and exclusivity. On this slide, I have provided the definition for both. So inclusivity is determined by conducting a study that involves pure target strains that shall be detected or enumerated by the candidate method. Alternatively, exclusivity is determined by conducting a study that involves non-target strains, which are potentially cross-reactive, that shall not be detected or enumerated by the candidate method. So ultimately, the goal of performing these studies is to ensure that the method will detect and or enumerate the microbes of interest and nothing else. And some of you may be wondering, where do I start with this? How do I know what should be tested for when it comes to inclusivity and exclusivity? Well, luckily, those AOAC SMPRs that I mentioned typically provide a required and suggested list of these microorganisms for each method. And I have provided an example screenshot from the Cannabis Salmonella SMPR here. And as you can see, the inclusivity requires several subspecies of Salmonella, while the exclusivity requires other microbes that could potentially be similar enough to be detected by a method if it was not developed properly. The next part of method development that I would like to review is detection limits and assay sensitivity. And this is a really important part of the method development process because it is going to be what determines whether or not laboratories are able to see and identify the analytes of interest at the required concentration. So when we think about analytes like Salmonella or Shiga E. coli, where they are deemed presence-absence analytes that have a concentration limit of 1 CFU per gram, can the method actually see that low? Can the method truly see down to 1 CFU per gram? And how do you determine that it was able to do that? And that's part of this method development process is, can we certify that our method is sensitive enough to be able to detect the microbe at 1 CFU per gram, which is an extremely small amount of microbes. And so for these two factors, I want to go ahead and break the method development out into two separate categories, being the qualitative and the quantitative. So for qualitative methods, we're looking at methods that just say, can I see this analyte? You don't have to say an exact concentration, just is it there? And for this, AOAC SMPRs for microbes have provided a definition for the probability of detection, or POD, which is defined as the portion of positive analytical outcomes for the qualitative method for a given matrix at a given analyte level or concentration. And this is what's going to play into assay sensitivity and the method's ability to positively identify the microbe at that required limit. So for example, ensuring that Aspergillus can be seen down to 1 CFU per gram in order for the laboratory to confidently say that it was absent from a sample. In turn, you have quantitative methods where you have to provide an actual concentration. You have to say this has 50,000 CFU per gram or 20,000 CFU per gram, whatever it may be. And in this, the SMPRs from AOAC for microbes have a similar definition for POD, but they also include definitions for limit of detection and limit of quantitation. And the reason being is that in addition to ensuring that the assay has the sensitivity necessary to positively identify that microbe, the method must also be able to successfully quantify the amount of that microbe that is present in the sample. So as a result, the SMPR has defined the limit of detection as the analyte concentration at which the POD is equal to 50%, and then they've defined that LOQ, the limit of quantitation, as the lowest analyte concentration that can be quantified with an acceptable level of precision and trueness under conditions of the test. And these LODs and LOQs are really an important part of ensuring that the laboratory can see and quantify those microorganisms at the required amounts. Because if the LOD is too high or the LOQ is too high, then they may be out of compliance. If their regulatory limit is 1,000 CFU per gram, but the LOD is 500 and the LOQ is 2,000, the laboratory can't confidently see that analyte at the required concentration, and so the method is no longer applicable for what the requirements are. Since I am running out of time, this is going to be the last portion of method development that I cover. However, I would be happy to chat about some of the other considerations offline if you have additional questions. So now we are getting into matrix validation, which can also be defined as matrix claims. And this is an important part of the process because it ensures that the method is truly applicable for the matrices that are going to be tested using the assay. And with matrix validations and matrix claims comes understanding how the matrix is going to impact the assay and what matrix interferences may occur. So depending on the method that's being developed, there may be interferences that occur with some edibles and not others. For example, if you have a gummy that is produced using pectin versus gelatin, does that impact the assay's ability to work properly? How do you ensure that the assay is working properly when one was made with pectin and one was made with gelatin? Those are two different substances, therefore would those be two different matrices? And really this is where cannabis makes things a little difficult because new products are being created constantly, and there really is no way that a method developer could possibly validate their assay for every available matrix. And as a result, when developing methods, there are specific parameters for matrix claims and matrix validations. And I've included a screenshot from one of the AOAC microbial SNPRs for cannabis that is going to show those guidelines. So what you can see here for that first one there, it says broad range of cannabis and cannabis products. You can see that the number of matrices is 15 with a minimum of 3 matrices per category, and the number of categories, a minimum number of 4 categories. And while I agree that it's just not realistic for a method developer to possibly validate their methods in every applicable cannabis matrix, this is really where the laboratories are going to be coming into play, because anytime they're using a validated method, they need to be doing the proper verification checks to ensure that that method is not being impacted by the matrix and that that assay still works properly even though that matrix may be different. And with that, I believe that we are out of time, but I did want to say a special thank you to Cannabis Science and Technology and AOAC for the opportunity to present this information. I've also included my contact information below, so please feel free to reach out to me if you have any questions.